Hello everyone, welcome to the new session of quantitative inheritance. So this is the first part. Okay, so let us start this session. Okay, so quantitative inheritance. What is the meaning of quantitative inheritance? So that is inheritance that is controlled by the multiple genes, right? Multiple genes. In the case of Mendelian inheritance, we have seen that the inheritance was controlled by single gene having two or more than two alleles. Okay. So the phenotypic traits of the different organism may be of two kinds. So we can have either qualitative or a quantitative, right? So what does this mean? The phenotypic trait in the different organism, like in pea, like in uh, any animals, okay, like in snails, okay, wheat, skin color. So there are so many organisms, right? And their phenotypic traits, how this phenotypic trait is controlled. So that can be studied in two ways. Either they can be a qualitative or a quantitative, right? So what is the qualitative traits? That are the classical Mendelian trait, that follow the classical Mendelian trait which we have already studied, right? And that is a kind such as a form round or wrinkled seed that we have seen in the case of pea plant. Okay, so we have seen how the round and the wrinkled seeds of a pea plant inheritance from one generation to another generation, and they follow the classical Mendelian trait, right? Structure. So horned and hornless condition in the cattle. So some cattle are having the horn and others are hornless. So how does this occur? Right? So these are the examples of the qualitative traits that follows the Mendelian traits. And this qualitative traits are controlled by single gene having two or more alleles. So in the case of round and wrinkled, we have seen that round, that is denoted by capital R. Okay, and if the seed color, we are also considering the seed color, then it will be yellow. Suppose yellow, round, so it is denoted by capital R, and wrinkle, that will be denoted by small r. So in this case, we can see that there is a single gene, and it is having two alleles, capital R and small r. Okay, so this is one of the example for the qualitative traits. Now, each qualitative traits may be under the genetic control of two or many alleles of a single gene. Okay, single gene may have two or more alleles. Suppose it is having two alleles, than in the previous case, round and wrinkle. That is the shape of seed. Okay, so each qualitative trait may be under the genetic control of two or many alleles of a single gene with little or no environmental modification to obscure the gene effect. So there will be no environmental effect on the this particular gene, no environmental effect, right? So what will be the effect? That will be purely concerned on the allele which is carrying, if it is carrying the capital R or if it is carrying the small r. So it depends upon the phenotype, depends upon the type of allele only. The organisms possessing qualitative traits have a distinct, separate phenotypic classes and are said to exhibit the discontinuous variation. So the organisms that are following the qualitative traits, the traits which we have discussed like just now, that single gene may have two or more alleles and there will be no environmental effect on it. Okay, and it governs a character or trait, right? So the organism possessing the qualitative traits have a distinct, separate phenotypic classes and are said to exhibit the discontinuous variation. Okay. Okay. So the quantitative traits, however, are economically important, measurable phenotypic traits of a degree such as height, weight, skin pigmentation, susceptibility to the pathological disease or intelligence in man, amount of flowers, fruits, seeds, milk, meat or egg produced by the plants or animals. So these are the different quantitative traits example, right? So we are going to study in this particular session mainly on the quantitative inheritance. So we will consider the quantitative traits. Okay. So the quantitative traits are called metric traits. What does this mean? So they do not show clear-cut difference between the individuals and forms a spectrum of phenotypes. Means there will be a different range of individuals, different range of phenotype, and that is called as spectrum of phenotypes, right? So they do not show the clear-cut difference like in the case of qualitative, capital R, small r, there will be a clear-cut difference phenotypically, round, wrinkled. So this one is totally different from the wrinkled one. Round is totally different from the wrinkle. But here in this case, there will not such a clear-cut difference, okay, which blend imperceptibly from one type to another to cause a continuous variation. So this shows the discontinuous variation. Discontinuous variation. Which one? Qualitative inheritance. Qualitative inheritance shows the discontinuous variation and there is a clear-cut difference between the organism phenotypically, right? But here you will have different range and the phenotypes are blend with each other. So there will be not so different that you can clearly say that this phenotype is totally different from this, right? So there will be a range of phenotype. Okay. So this means that they blend imperceptibly from one type to another to cause continuous variation. Very, very important. Okay. So in contrast to the qualitative traits, the quantitative traits may be modified variously by the environmental condition and are usually governed by many factors or genes. So here we will have a number of genes, not a single gene, but here it is governed by single gene. No involvement of environmental factor, right? And number of, in the case of quantitative, there are number of genes involved and there is involvement of environmental factors. So this is an example of continuous variation. So there are certain differences between the qualitative and quantitative inheritance. Qualitative, it is controlled by a single gene having 2 or more alleles. In the case of quantitative inheritance, it is controlled by a number of genes. Okay. No involvement of environmental factor in the case of qualitative inheritance. But in the case of quantitative inheritance, there is an involvement of environmental factors. As we can see in the case of skin color, right? Environmental factors are responsible for the change in the skin color. Next is the difference is the discontinuous variation in the case of qualitative inheritance, whereas in the case of quantitative inheritance, continuous variation can be seen. Okay. Each contributing such a small amount of phenotype that their individual effect cannot be detected by the Mendel method but by only statistical methods. So there are number of genes governing one character. And all you know that the gene will have different alleles also. They will have different types of alleles. So there will be, you can have a range of phenotype here. You will have a range of phenotype because there will be the different combinations of alleles. All the time you will not have the same type of combination of so many genes, right? And that is why they will have a different range of phenotype, also called as spectrum of phenotypes, right? So each contributing small amount of phenotype. So this particular gene will maybe have 2 or 3 types of alleles, right? So in one type of character, it can have a dominant, whereas in the other case, it will have a recessive. So there will be a difference. Now again, for this particular gene, they can have a number of alleles, right? And for the third and so on. So now you can imagine how many types of combination they can have in involvement of so many number of genes, right? So this cannot be detected by the Mendelian method. So for knowing this, how it works, we have to follow the statistical method. Okay. So such genes which are non-allelic and affect the phenotype of a single quantitative traits are called polygenes or a cumulative gene. So this is also showing the cumulative effect because the number of genes are involved, and this particular character is coming from the involvement of all the genes. So that will be the cumulative effect of the gene or polygene. Okay. The inheritance of the polygenes or the quantitative trait is called the quantitative inheritance. Multiple factor inheritance, multiple gene inheritance, or the polygenic inheritance. So there are multiple names given for this particular type of inheritance. So do not get confused if it is asked about you studied the quantitative inheritance, but in the examination it is asked about the multiple gene inheritance. That is the same thing, right? And the polygene, that is also the same thing. And in case if it is asked the multiple factor inheritance, what does this factor mean? The multiple gene. So this is the same, right? And the polygenes means many genes are involved. So this all are same thing. So do not get confused if it is asked in the examination, right? So the genetical studies of the qualitative traits are called as qualitative genetics. So we could have a different branch of genetics that is called as qualitative genetics that follows the qualitative traits, that follows the qualitative traits. The quantitative inheritance have following characteristics. The segregation phenomena occur at an indefinitely large number of genes loci. Okay, so there are number of genes involved. So all the genes are located on the different, all the genes are located on the different loci. So suppose I'm taking one example. This is one particular chromosome, right? Paired form. This is the first chromosome. Okay, we are having 1, 2, 3, 4 genes involved, right? And we are having again another chromosome. Say, this is, I'm just taking an example to understand. Say chromosome third chromosome, right? This is the third chromosome. And suppose I'm taking one more chromosome, say 7th chromosome. 7th chromosome, right? So this different loci, they are located at the different loci. So I'm taking the chromosome number 1. So it can be located on here. Suppose it is located here, right? This is loci 1 of chromosome. Okay. Gene 1, we can say. Gene 1, right? And gene 2 comes to the gene 2. It is located here, suppose. Gene 2. Gene 3, it is located here. So it is gene 3, right? And gene 4, coming to the gene 4, it is located on the 7th chromosome. This is the gene 4. Now, this gene, and this govern one character or trait. Okay. So the segregation phenomena occur at an indefinitely large number of genes loci. So we are having large number of gene loci. If a substitution or allele occur in a gene locus, then such allelic substitutions have a trivial effect, means no effect. Okay. The gene of a multiple trait have a different biochemical function but similar phenotypic effect. So we will have a similar phenotypic effect, and if they have a different biochemical function but the similar phenotypic effect can be observed. Therefore, the phenotypic effect of a gene substitutions are interchangeable. Blocks of genes are bound together by inversion and transmitted as a unit from inversion heterozygotes to their progeny, but such blocks are broken up by the crossing over in the insertion of the homozygote. So what happens? Sometimes there is an inversion, sometimes there is a crossing over. So when there is a crossing over or inversion means a gene is deleted from here and it moved to the other place. So that is the inversion. So blocks of genes are bound together by inversion, right? always they are bound together by inversion and transmitted as a unit from inversion heterozygotes to their progeny. Okay, and such blocks are broken up by the crossing over in insertion homozygotes. The polygenes, okay, the polygenes have the pleiotropic effect that one gene may modify or suppress more than one phenotypic trait. So one gene either can modify, it can modify or suppress the effect, the phenotypic effect of a trait. Phenotypic trait or a character, right? This is one gene. So this is the showing the pleiotropic effect. A single allele may do only one thing chemically, but may ultimately affect many characters. Single gene can have one chemical, can have only one chemical. But when there are multiple genes and they are having different alleles, then they will have a different combinations. So they can affect many characters. They can affect many characters. Many characters. Okay. The environmental condition have considerable effect on the phenotypic expression. Polygenes for the quantitative traits. So in the case of quantitative traits, environmental factors have a considerable effect on the phenotypic expression. For example, height in many plants, corn, tomato, pea, marigold, is genetically controlled quantitative trait, but some environmental factor like soil, fertility, texture of and water, the temperature, the duration of wavelength of incident light, the occurrence of the parasite also affect the height of these plants, right? So if you provide a good quality soil, the height of the plant will be good because it's getting all the nutrients. Fertility, soil fertility is better, texture and water availability, very important. If the water availability is more, the plant will flourish. And if the water availability is less, the plant cannot grow to a great height, right? Temperature also affect the height and other characteristics of the plant. Duration of wavelength of incident light, and yes, the parasites. If the plant is affected by the parasite, then it will do not have a proper growth. The height will be stunted, so the plant can be dwarf. So there are different effect of the environmental conditions depending upon how the environmental factors affecting the plant, which of the factor the plant is not having, and which of the factor the plant is getting at one particular time, right? So this environmental factor totally determine the effect, it has an control on the phenotypic expression of the polygenes. So let us take an example of the quantitative inheritance. Okay, so kernel color in wheat. We will have an example of the kernel color in wheat. So Nelson, Abel and the East gave first significant clue of the quantitative inheritance by taking an example of wheat. So they have worked a lot on this particular wheat and they have a certain results. Okay, so they crossed a strain of red kernel wheat plant with another strain with having the white kernel. So two contrasting, they have taken, that is the red kernel wheat plant and the white kernel wheat plants. And you can see here R1, R2, R2, that is showing the dark red, and R1, small r1, small r1, small r2, small r2 showing this a white. Okay, so here we are having, this should be white, and here we are having medium red in the F1 generation, F1 generations. And what is the combination? The combination is R1, small r1, r2, small r2, right? So this is the individual that we get in the F1 generation. Now we will take two plants, one acting as a male, okay, and another acting as a female. So there will be egg in the endosperm, in the embryo sac, and sperm. Sperm as in pollen grain, the male gamete. So here we can have so many combination, capital R1, capital R1, capital R2, capital R2, like the parent one. This is the parent one. And here also this is the parent one, right? And there we can see different combinations like this. So we are having 1, 2, 3, 4, 5 range of combinations. Okay, color variation in wheat we can see, and we can see this skin color. So at the end we can see here of the graph, this is the showing graph, this and this showing the parental, this is also showing the parental form. Okay. So grain from the F1 was uniformly red, but of a shade intermediate between the red and white, the parental generation. So here we can see it is a medium red. It is a medium red. So we are taking one as a male and one as a female plant, and we can have such a combination. We can have so many combinations. So this might suggest incomplete dominance, but F1 offsprings were crossed among themselves. The F2 zygotype showed 5 different phenotypic classes in a ratio of 1:4:6:4:1. So we are having these 5 different combinations. So nothing that 1 of 16 of the F2 was an extreme in the color as either of the parental red or white. Okay, so we are having 1 of 16, and this is also 1 of 16 as a parental one. Right? This is also another parental one, right? So they theorized that 2 pairs of genes controlling the production of red pigment while operating in this cross. So 2 pairs like R1, R1, R2, R2, and small r1, small r1, small r2, and small r2. 2 pairs of red pigment. Okay, and 2 pairs of white pigments are controlling this cross. Each gene was supposed to contain 2 alleles. So one allele produces a given quantity of the red pigment while its counterpart did not produce any pigment, right? So all alleles were equally potent in the production of the lack of production of the pigment. So one is producing the pigment while other is not producing any type of pigment. But there will be a different effect in the absence of not producing a pigment. So we can have a different variation of color in the progenies, in the F2 generation. Okay, so we can see here 1/16, that is the red, this one, 1/16. Okay, 4/16, that is the dark, 4/16, that is the dark. Okay, 6/16, that is the medium, 6/16, that is the medium. This is the medium, this is the dark, this is the red, and this one is 4/16, that is the light, and 1/16, this 1/16 is the white. Okay, so if we symbolize the gene for red with the capital letter A and B and their alleles resulting in a lack of pigment production by A and B, we can illustrate the result of this cross as follows. So we can have so many combinations. Okay. So this is all about this session. We will have one more session to understand some more example based on the quantitative inheritance. Till then, thank you very much.